So hello friends, so today we'll discuss about the previous year questions. So the first question here is the PCR is used to, okay. So first is detect the target plasmid to amplify the small amount of DNA and to seal the content of DNA and to cleave the bacterial plasmid. So always remember, so PCR is nothing but it is a polymerase chain reaction and it was discovered by Hazen and the Carl Mullis. And here regarding the options, the correct answer will be amplify the small amount of the DNA. Okay, so it always, so a simple or a small amount of DNA, it is converted into 1,000 and millions of copies of the DNA. Okay, so that is the main aim of the polymerase chain reaction. So here the option is amplify the small amount of DNA. Okay. So next, second question is, so tropical spastic paraphyresis is caused by. So here it is a straight question. So the option given here are the first is Epstein-Barr virus and human T lymphotropic virus, HIV and hepatitis B. So here the option is human T cell lymphotropic virus, and you want to know some of the points about that. First, it causes an tropical spastic paralysis and adult T cell leukemia or lymphoma and cutaneous T cell lymphoma and autoimmune manifestations such as uveitis and arthropathies. So again, this has been asked in previous year. Okay, the cutaneous T cell lymphoma, so it is like some of the clinical history they have asked, and what is an organism. So that is a human T cell lymphotrophic virus. Okay, so coming for third question. Okay, this is a clinical question. Here the female presents with complaints of pruritus, vaginitis and offensive yellow-green discharge, and there is a dysuria and dyspareunia. Okay, so local examination, there is a petechial hemorrhages and strawberry mucosa, and this is a term. Intracellular edema will be there, so it is like chicken-like epithelium. Okay, so here the option given are bacterial vaginosis, Trichomonas vaginalis and Candida albicans and Entamoeba. So here one clue is they have given clearly about that is in strawberry mucosa. You can make up that is a strawberry vagina or cervix. You can easily know it is in Trichomonas vaginalis. And the second point you want to know here is the another term, it is in chicken-like epithelium. Okay, so here you want to remember the chicken-like epithelium, and that is a new term over here. Okay, so coming for if here trichomonas usually causes an yellowish green discharge, whereas if they have given the bacterial vaginosis, they will be giving like a fishy odor, or what is that, if test is positive. Okay, the main feature is it is a fishy odor of discharge. If they are given Candida albicans, I'm sure they will be given a curdy white discharge. Okay, this is just to differentiate all the form of the discharges, but here they have given clearly it is an yellowish green discharge and strawberry mucosa, and the only term here the extra is chicken-like epithelium, which is caused by Trichomonas vaginalis. And this question has asked in recent years only, and this is a linear syndrome which is caused by the bacteria that is in Fusobacterium necrophorum. It usually causes in thrombophlebitis. The friends, in my YouTube channel there is a video for these syndromes alone. Okay, so I have like compiled all the syndromes of microbiology. You can go through that. Okay, so I will post the link over my in this video, and just go through that. So next question. Okay, so all are true about Aeromonas except it causes a blue leg disease in frog and Aeromonas is susceptible to cefotaxime and ciprofloxacin, and hemolytic uremic syndrome is caused due to production of enterotoxin, which is similar to Shiga toxin, and it produces an endotoxin. The answer here is, so it causes blue leg disease in the frog. Okay, so it usually causes a red leg disease. So okay, it causes a red leg disease. Some important points you want to know about this Aeromonas. It is an gram-negative bacilli, and it is a motile with single polar flagellum. And in McConaughey agar, it shows an LF, that is an lactose fermenting colonies. Catalase oxidase is positive, and decarboxylase test, at this point and all no need. Okay, so just remember, so decarboxylase test utilizes only arsenide, and both is not stimulated by sodium chloride. Okay, so here just remember, it is a gram-negative bacilli, first point, and it is a motile organism, second point. Third, McConaughey agar shows a lactose fermenting colonies, and fourth, it is an oxidation catalase positive. Fifth point, it usually causes a red leg disease. Okay, so these are the main points you want to remember in this question. So thank you, friends.